In this class, we are going to study about the cellular structure or the properties of the cellular components of the fungi. So, in fungi, we have already studied about the hyphal structure. So, hyphal structure were mainly forming the mycelium or the main body of the fungi. But in this class, we are going to study about the individual cells which make up the fungi. So as we know that a cell is composed of many cellular organelles. So we know that fungi is an eukaryotic category. So it is but obvious that fungi cell wall would be just like a typical eukaryotic cell. So in this case, fungi contains almost all the normal cell organelles which are membrane bound, not just like prokaryotes where only mesosomes were formed. So here this fungi cell would contain almost all the cellular components of a typical eukaryotic cell. For example, the endoplasmic reticulum, the nucleus or the vacuole or the lysosomes and various other cell organelles. But there is one very important difference between a plant cell and a fungal cell, and that is the presence of plastid. So in a plant cell, plastid is present, and in fungi cell, the plastids are absent. So what are these plastids? So these plastids are generally of three types. For example, in plants we have three types of plastids, like chloroplast, which contains the chlorophyll, the chromoplast, which contains other pigments like xanthophylls, and also the leucoplast, which does not contain any specific pigment, that is, it is colorless. So this fungi cell does not contain these different types of plastids. So it is a very important difference between a typical plant cell and also a typical fungi cell. Apart from these plastids, when we compare a normal fungal cell with a normal eukaryotic cell, we normally find that almost all the components of a typical eukaryotic cells are present in the fungal cell. So that is why these are eukaryotic cellular structure. These have eukaryotic cellular structure. Therefore, they have almost all the organelles of a typical eukaryotic cell. And what is not contained here? And that is plastid. So plastids are normally the pigment containing cellular organelles. So that is the first thing about the cellular structure of the fungi. Now in fungi, we know there is something called as cell wall, which is also present in a typical plant cell. So a plant cell wall is normally composed of cellulose, and which is a polysaccharide, by the way. And here also there is a presence of something called as chitin, which is also a polysaccharide, but some amine components are also present over here. So this cell wall present in the case of fungi is composed mainly of chitin, which is also a polymer. So this chitin is also known as fungal cellulose. So in normal plant cell, we know that cellulose is present in the cell wall. Here also the fungal cell wall will be mainly composed of this chitin or fungal cellulose. And also some other components are present. For example, polysaccharides of other types like other than cellulose, like amylose or amylopectin or any other such polysaccharide. Apart from that, some proteins and lipids and commonly oils are present in the cell wall. So these are the component of a typical fungal cell. But fungi has various classes. For example, Phycomycetes or Ascomycetes or Basidiomycetes. So when we say that a fungal cell wall mainly composed of fungal cellulose or chitin, then it is not true for every category of fungi. For example, there is a category of fungi which is called as Oomycetes. So this Oomycetes has certain components of its cell wall which are mainly composed of cellulose. So we can say that in the case of Oomycetes, their cell wall is mainly like or very similar to the plant cell wall, because it is also composed of mainly cellulose apart from the other components. So some categories of fungi also have this chitin and cellulose in almost equal proportions, and in some cases like oomycetes, actually the cellulose is the major component of the cell wall, not the chitin. But in the majority of the fungi, chitin is the main component of the cell wall, and only in oomycetes this cellulose is the predominant material in presence in the cell wall. Now this chitin is also a polysaccharide, and it is mainly formed of N-acetyl glucosamine. So we can also say that acetyl glucosamine, but normally that is fully named as N-acetyl glucosamine, and it is also a polysaccharide. But here we can see that amine. So this group makes it a heterogeneous group. So it is not a totally homopolymer, because there is something called as amine group is present over here. So we can say that some amino acids are attached over here, because this amine group comes from a normal amino acid. So although it is a polymer, but still it is containing some amino acid components, or we can say peptide components also. So this is what exactly chitin is, and we know that cellulose is a polymer of glucose. So here comes the difference between the fungal cellulose and the real cellulose of plant cells. So the cellulose of plant cell is mainly a polymer of glucose, but here it is a polymer, or it mainly composed of acetyl glucosamine, and here amine means some amino acids are present over here. So now we come to two vesicular shaped structure or membrane bound structure, which are called as chitosomes and lomosomes. So these have their specific location and function. So these lomosomes are normally present between the cell wall and the cell membrane of a fungi, and these are mainly associated with the cell membrane of a fungi. So these lomosomes are also present in some bacteria. So these are not just a component of fungal cell walls or fungal cell membranes, but these lomosomes are also present in certain bacilla, which is a category of the bacteria, which was the rod shaped bacteria, which is normally called as bacilli or bacillus. So this lomosomes is also present over there, but here these are membrane bound vesicles, which are normally present between the plasma membrane or the cell membrane, or we can also say that as plasma lemma. So between the plasma lemma and cell wall, these are vesicular structures which are present. Now they have two very important functions, which is vesicular transport, and also these increase the surface area. So these are the two functions of this lomosomes. So the vesicular transport involves transport of certain materials. For example, it could be some chitin materials which are being transported, or various other materials which are packed in the forms of small sac-like structure, which are called as vesicles. And apart from various types of vesicles present in a cell, we are going to study about various components and which are the materials which are normally packed inside the vesicles in our cell chapter, that is chapter number 8. So we'll study about more, or we'll study about these vesicles in detail in our cell chapter. So a normal vesicle could contain some proteins or carbohydrates or fatty acids or any other such lipids. So these vesicles are mainly transport sacs. So these help in the vesicular transport. So it could be from the cell towards the cell wall, or from any part to the other part. So this is the function of lomosomes, and these also increase surface area, could be for excretion or absorption. So we know that fungi are a heterotrophic category, that is, these cannot synthesize their own food. So obviously these would have to be dependent on some other category or some other organism. So in that case, fungi is also a decomposer. So it can absorb certain material which are being decomposed, and for that absorption to occur, a very large surface area is required, because large surface area means more diffusion or rapid diffusion. So easy absorption of materials could occur, and that is why this lomosomes is increasing the surface area. Now there is some other vesicle like structure which is present near the ends of the hypha, and these are called as chitosomes. So we can know from the name that it is chito. So the name suggests that it has something to do with the chitin, and we know where is chitin located. So chitin is the cell wall, or chitin is mainly contained inside the cell wall. So these chitosomes are small vesicles, and obviously these vesicles are located in the cytoplasm, and these contain the wall forming material, and we know what is the wall forming material, that is the chitin. So these are present near the hyphal tip of the cytoplasm or the cell or the hypha, and contain wall materials, and as the name suggests, these cell wall materials are mainly chitin in the majority of the fungi. So these two are very important vesicular structure present specifically in fungi. So obviously there are certain other types of vesicles also, but we don't have to study about all those types in detail, but these two are the most important vesicle or vesicular like structure, we can say, that are present in the case of fungi cell. Now we have to study about some other features of a normal fungi cell. For example, in a normal eukaryotic cell, for example in the case of plants, there is a very large vacuole, but here a small vacuole would be present. So this is one difference between a typical eukaryotic cell and a typical fungi cell. And apart from that, the normal nucleus occupies the central or the major space of a typical eukaryotic cell, but not in the case of plant cell. So the nucleus in the case of fungi is much smaller than a normal or a typical eukaryotic cell. So these are the two differences between a typical eukaryotic cell and a fungi cell, that is the nucleus and the vacuoles. Both are smaller as compared to a normal eukaryotic cell. For example, in an animal cell, a nucleus occupies a very prominent space, but here a nucleus is small. And in a plant cell, the vacuole occupies 40 to 90% of the volume of the cell, and here the vacuoles are smaller. So this is a very important point of difference between eukaryotic cell and a typical fungi cell. Now you must have studied about a cell organelle which is called as Golgi body or Golgi apparatus. So these Golgi apparatus are commonly composed of many flattened sac-like structure, something like this. So a normal Golgi apparatus in an animal cell is something like this, and these individual structures are called as cisternae. So these dictyosomes could also be said to be as cisternae. So in this case, that is in the case of fungi, these Golgi apparatus, or we can say dictyosomes, are unicisternal. That is, these do not contain so many cisternae. These contain only one cisternae. So in the case of fungi, the dictyosomes are normally unicisternal. But the diagram that we just drew, that diagram, such type of Golgi apparatus would be found in animal cells. But here in the case of fungi, the Golgi apparatus or the dictyosomes are unicisternal, that is only one cisterna are present. Now every organism needs some form of stored food, and likewise fungi also needs some stored food, and that is mainly in the form of glycogen and oil. And you must be remembering that glycogen is the major food storage food form in the case of animals. In humans also, this glycogen in humans also, this glycogen is present in our liver and also in our muscle cells. So in the case of fungi also, the food is mainly stored in the form of glycogen. So glycogen is a storage carbohydrate, and this oil is a storage lipid. So lipid is stored in the form of oil, and carbohydrate is stored in the form of glycogen. So this oil and glycogen are storage structure or storage form of food in the case of fungi. Now there is a very important point which is to be understood in the cell division chapter. So in the cell division chapter, we will study about a nuclear division. Suppose this is a typical cell and this is the nucleus. So when the cell divides or just before division, the nuclear membrane breaks down. In a typical mitosis or meiosis case, this nuclear membrane breaks down before a cell division. But here in the case of fungi, this does not occur. That is, nuclear envelope does not break down during nuclear division. So in a normal cell, the nucleus will divide or the nuclear envelope will break down. But in the case of fungi, this nuclear envelope or nuclear membrane will not break down. It will remain like this. In a normal eukaryotic cell, when the cell undergoes nuclear division, or that is also called as karyokinesis. So karyokinesis is the another word for nuclear division. So in the case of karyokinesis, this nuclear division does not involve this nuclear membrane breakdown. So this is what is happening in the case of fungi. So this will remain like this will not occur. Apart from that, there is a very important structure which is called as spindle fiber. So this spindle fiber, these are structures which are normally formed between the poles, something like this. So these are the spindle fibers which are formed outside the nucleus in the cytoplasm of a typical eukaryotic cell. But this is not happening in the case of fungi. This spindle formation will take place, or this spindle fibers will form, but these will be formed inside the nucleus. So obviously these spindles are being formed, but these are formed inside the nucleus. This is the difference between a spindle fiber of a typical eukaryotic cell and in the case of fungi cell. So we will study about this karyokinesis, nuclear envelope breakdown, this spindle fiber formation in the chapter cell division, that is the 10th chapter of standard 11th. So these will be studied in detail in that chapter, but here we must have to remember that the fungi nuclear division does not involve nuclear membrane breakdown, and also the spindle fibers will be formed inside the nuclear membrane. That is, we can say that these spindle fibers are being formed inside the nucleus, not outside the nucleus, just like in the case of a plant cell division or an animal cell division. So this is the very important point of difference between the nuclear division of fungi and plant or animal. So this is what is the difference between plant and animal and fungi cell division. Apart from that, there is something called as meiosis and mitosis. So meiosis and mitosis are also to be studied in detail in chapter number 10, but here we must remember that this meiosis is a sort of reductional division and this mitosis is a sort of equational division. So in a fungi, these both could occur. So in meiosis or mitosis, in either of these cases, the nuclear envelope will not break down, and also the spindle fibers will be formed inside the nucleus. So whether be it mitosis or meiosis, the spindle fibers are not getting formed in the cytoplasm or outside the nucleus. Now there is something called as volutine or metachromatic granules. So these granules are also present in some bacteria. For example, the bacteria that causes diphtheria, that is the Corynebacterium. So these are also present in those cases. So what are these? These are basically a storage sort of a structures which contain polymerized phosphate. So we know what is phosphate. This is the formula for a phosphate ion. So these structure, that is the volutine or the metachromatic granules, these contain these phosphates in a polymerized form. So we can say that these are storage bodies of polymerized inorganic phosphate. And what is the purpose? So the purpose is this acts as a reservoir or as a reserve of energy and phosphate, because we know that ATP, that is adenosine triphosphate, is the energy currency of the cell, and for ATP formation to occur, phosphate group should be there. So we can say that this phosphate or polymerized phosphate reserve, which is present in the case of metachromatic or volutine granules, is a sort of reserve or a storage. So these are used whenever required. Now there is another property which is called as metachromatic property. So this metachromatic property is nothing but being able to pick up a color. For example, in the presence of methylene blue, so this is a dye. So these granules or the volutine granules, these appear red when we use this dye, that is called as methylene blue. So this is a dye, or we can say a coloring agent. So in the presence of this coloring agent, this metachromatic granules or volutine granules, these appear as red. So because of this color property or because of this ability to pick up the color, these volutine granules are also called as metachromatic granules. So this was about the more common or the various simple features of a typical fungal cell. So after this cellular structure, and also we have studied the hyphal structure, we are going to study about the various methods of reproduction in fungi.